So we'll be talking about Plasmodium vivax, the life history of Plasmodium, life history. So before I begin with the life history, I will talk about the two hosts which are there in the life history. So the first one is the man and another one is the mosquito. In man, malaria is caused, whereas the mosquito is harmless. Mosquito contains, or we can say that it carries the sexual phase, and it is also known as the final, or we also call it as the primary host or the definitive host. So these are the other names to the host, right? So another we talk about man, where asexual phase is taking place. Asexual means we'll be talking about how binary fission is going to take place here. It is the asexual phase, right? Asexual phase, and we call it as the secondary or the intermediate host. If we have to differentiate between, before I begin with the life history, let me just differentiate you with the male and female anopheles. So I think, as you all know, it is only the female anopheles which carries this parasite. So how do we differentiate between the male and the female? Male is, if you talk about their antennae, they are very bushy. Whereas in case of females, they are not that bushy. So comparatively lesser bushy. So I think by visualizing the antenna, you can, so on the basis of its antennae, you can differentiate between male and female anopheles, right? So this is just the basic information about Plasmodium vivax life history. Now I discuss in detail the life history, how exactly the asexual and the sexual phases are going to run in this particular life history. So life history is further divided into three portions: Schizogony, Gamogony, and the third one is Sporogony. Schizogony is the phase of growth. It is the phase of growth. And another thing, the asexual multiplication is taking place in case of schizogony. So this is first phase of growth, another asexual reproduction, and the third one is that it occurs only in man, right? So this is regarding the schizogony. Then next we have gamogony, where this is considered as the sexual phase. Sexual multiplication takes place. Here it starts in man and it is going to be completed in the mosquito. So this is about the gamogony. Gamo means gametes. So basically in this particular stage, we will see the gametes are going to be formed. And lastly is the sporogony. Sporogony, it is the phase of sexual reproduction again. Sexual multiplication, which takes place only in mosquito, right? So this is just a brief information about the three phases. So first I'm going to discuss about schizogony, right? So this schizogony is in two phases. Very important. The first phase is Exerythrocytic, or we also call it as the hepatic schizogony. Exerythrocytic or hepatic schizogony. And the another one is erythrocytic schizogony. Okay, if we talk about where this exerythrocytic schizogony takes place, so it takes place in the liver cells. Hepatic means hepatic cells, so it is in the liver. This takes place in the liver. Erythrocytes, from the name itself, erythrocyte, it takes place in the RBCs. So it is in the RBC, right? So these are the two phases under schizogony. So I'm discussing about the first phase of schizogony, that is of exerythrocytic schizogony. So I'm discussing first the first part, that is the exerythrocytic, or we also call it as the hepatic one, which is found in the liver. So this is again further divided into two types. First one is the preerythrocytic schizogony and post-erythrocytic schizogony, right? So first I'm going to discuss about the preerythrocytic. So basically this is the broad category. The habitat remains same, that is the liver cell, right? So under the preerythrocytic schizogony, it starts with the first portion, the exerythrocytic schizogony. It starts with the inoculation. So what do you understand by the term inoculation? Or it is infection to man. So how basically this infection to man takes place? How the parasites, they enter into the human blood. Parasites enter the human blood when an infected, so this is important here, when an infected, a female anopheles has to be infected when they are putting or inserting their parasite into the host. So female anopheles has to be, if the female anopheles is not infected, she is not the source of infection, right? So and female infected anopheles is very important. And why these female anopheles suck the blood? Normally male, they suck the plant sap. Male anopheles, they suck the plant sap. Why only females suck the blood? So the reason behind it, the females require, you can say they suck the blood for their egg production. So this is a very important reason behind it, why only female anopheles are sucking the blood, because this is basically, blood is the meal for whether it is infected, whether it is not infected, the blood of human, but they require it for the production of eggs. Okay? So it acts as a meal, we can say it that way also. Okay, now how the female anopheles is infecting the or inoculating into the man. So as we all know that anopheles has a piercing and sucking type of mouth parts. Okay, so piercing and sucking. So as the name is specifying, piercing. So first it is going to pierce into the skin and simultaneously it is going to suck the blood. So it is just like the needle. It is going to, with the help of its proboscis, it is going to inject those needle-like proboscis into the skin of the human being and take the droplet of blood, and at the same time it is also going to inject a saliva, right? So if that female anopheles is infected, so that parasites are going to enter into the blood of human at the same time. If it is not infected, automatically the life history is not going to continue, right? So here we can say that the saliva carries the parasites to human blood. But the condition for that has to be there, that the anopheles is infected, right? Now, the infective stage, the infective parasite is known as, or we can say this infective stage also. So this infective parasite is the sporozoite. So this is the starting point where our preerythrocytic schizogony is going to start, right? So before beginning with the cycle, how exactly the sporozoite looks like? So basically it is sickle shape. So it is like this, somewhat like this. It has a slender body, as you can see, slightly curved. Okay? Because if you see its edges, they are not straight, they are curved. It has a sickle shaped body. Okay, so one thing is your sickle shaped body. Another one, if we talk about how long is it, obviously I have made it a longer one, but otherwise how long it is 50 micrometers long. And it is enclosed by a pellicle. It is thin pellicle, or we can say a cuticle, and it is because of this cuticle it can maintain its shape. There in the broader part of the sporozoid is the nucleus, right? So the middle part is the nucleus. Other to it, the sporozoid has a very important feature of gliding, right? So it is because of this nature that it can glide in the blood, and at the same time, after half an hour, it can penetrate into the liver cell. So it has this gliding movement. Okay? Had it been, it did not have this gliding movement, it would have stayed at one particular position only. Okay? So movement, gliding movement. Okay. Now, this is the first stage where the infective stage of parasite, or you can say it is going to infect the human being. Next point I'll discuss, that is the how multiplication of parasites are going to take place. So how does it take place? The sporozoites, when the infected Anopheles has bitten the human being, so the sporozoites have entered into the body of the human blood, right? Now, it is going to, the first very important, immediately after sucking, or you can say injecting the parasite, the sporozoites into the human being, it is not immediately going to go to the liver cell. It is going to stay in the blood for around half an hour. For half an hour, it is going to stay in the blood, right? And then after half an hour, and again, very important thing here, when it is staying for half an hour in the blood of the human, right, it is not the source of infection, right? Even if some other mosquito comes and sucks up the blood, so it is not going to be the source of infection to some other human being, right? So it is only in the blood for half an hour, and then after half an hour, it is going to enter into the liver cell. Right. So how the liver cells look like? So the liver cells, we all know that they are hexagonal in its shape, right? Hepatocytes, they are made of hepatocytes. So now each sporozoid, because they are going to be n number of sporozoids. So these sporozoids are entering into the liver cell. So here each sporozoid are going to enter into different type of, or you can say different liver cells, but here I am just talking in respect to one liver cell, how it is affecting one liver cell, right? So this sporozoid, now you can see here with the nucleus, with the sickle shape, now it is penetrating into the liver cell. So within the liver cell, there is a nucleus. There is a nucleus. Sporozoid is entering. This is the first step now. Okay. Next, this next step is, you can see when the sporozoid is entering into the liver cell, it is still sickle shape. But the second stage, the nucleus remains there. Immediately it enters into the liver cell, it becomes rounded in structure. Okay, so this sporozoid has become rounded in structure immediately as it enters into the body, the liver cell. So we call this stage as, when the sporozoid finally enters into the liver cell, we call it as the cryptozoid, right? So it is cryptozoid, right? Then this cryptozoid, basically, if we talk about its ultrastructure, so it has a compact nucleus. It lacks vacuole. Okay, so it lacks cryptozoid. It lacks vacuole and pigment, which is basically the characteristic feature of a trophozoid cell. So, right? So this is basically, it has a nucleus, but it lacks vacuole and pigment. This is the second stage. In the third stage, what happened? The nucleus moves towards one side, right? And the cryptozoid, because within the cryptozoid there are cytoplasm also. So within this cryptozoid, this feeds on the cytoplasm and it becomes a rounder structured body. So again, this rounded cryptozoid, again, it has a nucleus in between. Okay, but now you can see the size has increased and the nucleus has been displaced to bit side to side. And now this particular site, we call it as the Preerythrocytic schizon, or we also call it as cryptoschizont. So that is up to you what you want to remember. Preerythrocytic schizont or cryptoschizont. So how cryptoschizont is found? It is formed when the cryptozoites feeds on the cytoplasm and it goes into this rounded structure. So this is the third structure, right? Now after this stage, what happens? It undergoes this cryptoschizont, it undergoes multiple fission. So this is the cryptoschizont, the third portion. You can see here it is the schizont, right? And it has a nucleus in the center. Next step, what happens? This step is going to undergo multiple fission. So what is going to undergo multiple fission? Cryptoschizont is going to undergo multiple fission. So here we can have another diagram which is going to be a bit bigger one and a nucleus, right? And now we can mention it as that multiple fission is taking place. Multiple fission is taking place, right? So after this, in this process, what exactly is going to take place? The nucleus, this particular nucleus which we have discussed in the cryptoschizont, that is going to break into, so nucleus is going to divide into many nuclei. So it is going to, during multiple fission, it is going to divide into numerous nuclei. So each nuclei here, which is very interesting, each nuclei is going to gather, it is going to, with each nucleus, there's going to be a cytoplasm. So basically, it is going to feed on this nucleus is going to feed one liver cell. So the number can be around 1000 parasites. So 1000 parasites in one liver cell. So we can imagine the amount of damage that is going to be in the entire liver. Next step after it, after the cryptomerozoite has been formed, the schizont, right? We call it as the schizont. The schizont and the liver cell is going to break up, right? So here we can have the structure where the schizont has broken out, right? And the liver cell, the wall of the liver is also up. So here, now what is going to get liberated out? Cryptomerozoites are going to be liberated out. The nucleus is still there. So these cryptomerozoites, they enter into the liver sinusoids. Before entering into the liver, they enter into the liver sinusoids. And what are these sinusoids basically? These sinusoids are small blood channels which have an incomplete wall. So small blood channels with incomplete, another thing that is with an incomplete wall. Once they have entered into the liver sinusoids, after that the cryptomerozoite is going to enter into the fresh liver cell. So now it has entered the cryptomerozoites. The cryptomerozoite have entered into the fresh liver cell. Fresh liver cell. Cryptomerozoite which has entered into the fresh liver cell to restart the Excerythrocytic schizogony, or we can say to restart the Preerythrocytic schizogony. So again we start with it. Cryptomerozoite is entering into the fresh liver cell. So what is entering? Cryptomerozoite is entering into the fresh liver cell. The process again remains the same that we had just discussed previously. Once this cryptomerozoite has entered into the liver cell, it again becomes a rounded structure as we just discussed in the previous phase, that is when cryptozoites were formed. So when this cryptomerozoite enters into the a fresh liver cell, it changes into the another form that is known as, the word meta comes here, that is metacryptozoid. Previously it was cryptozoid, right? So only the word new, that is metacryptozoid. So this term basically was given by Huff, Huff. Now this metacryptozoid, same as previously we had discussed, it is going to, this is the nucleus, and now this metacryptozoid is going to feed on the cytoplasm, and it is going to become rounded in structure. It is going to grow in size, and it is going to form the metacryptoschizont. Like in the previous one, we discussed it as the cryptoschizont. So here in the next, or you can say the second fresh liver cell when it is entering, we mention this as meta, right? So this metacryptoschizont is going to undergo the next phase, that is with the multiple fission. So here is the nucleus in the schizont. So it's the same way. This is the nucleus of liver cell. The schizont nucleus is again going to divide. Okay, it's going to undergo multiple fission, where it is going to form around 64 to 1000 metacryptomerozoids. So this is basically the another form which we call as the metacryptomerozoids. So how they are formed? They are formed when the nuclei of the schizont, nucleus, it divides to form many nuclei, and those nuclei are feeding on the cytoplasm. It forms metacryptomerozoids. So how many are they in number? If previously you remember, we discussed about the cryptomerozoids. They were around 1000 parasites. So here they vary from 64 to 1000 parasites. Now this metacryptomerozoids are of two types, right? So what are those two types? Macro, right? Only the word macro and micro is going to come in front. Rest is same. So it's not a very difficult term to memorize. Metacryptomerozoids. And another one is micro-metacryptomerozoids. So as the term is macro and micro, right? So macro means something bigger in size. So here they are larger but few in number, right? Whereas micro, they are smaller in size, but the number is numerous. So both these metacryptomerozoids, right? Both macro and meta, they are going to be again released into the liver sinusoids, as previously also we had discussed, right? And from liver sinusoids, either they are going to enter into the liver or into the RBCs, right? So basically, the habitat of micro-metacryptomerozoid is RBCs, right? Whereas macro-metacryptomerozoid, the habitat is liver. So after this stage, once the metacryptomerozoids have been formed and they are of two types, we come to the next phase where the schizont is going to be released out. Okay, so we've already discussed it's been released out. Released out how? The liver cell is going to break. So here you can see how the liver cell is going to be broken. So this is how the liver cell is broken, and micro and meta are going to be released. So both within this metacryptomerozoids, both macro and micro both are formed. So from here they are going to go into the liver sinusoids, and liver sinusoids, after liver sinusoids, either they will be going to the liver or they will be directly going into the RBCs. Okay. So next we talk about the cycle that we have discussed, right, starting from the sporozoid when it has entered into the fresh liver cell, up to this particular stage when the metacryptomerozoids are formed, merozoites are formed, it takes around 12 days. We call it as the pre-erythrocytic schizogony. So I had discussed it with you.